We are fortunate to have Dr. Ashley Beasley Green provide our next lecture. Dr. Green is currently a chemist at the National Institute of Standard and Technology, and her research primarily focuses on the standardization of protein measurement and science. Dr. Green received a Bachelor of Science degree in biochemistry from Spelman College in 2004, followed by a PhD in pharmacology from Johns Hopkins. Dr. Green completed a postdoctoral training at NIST. Please enjoy today's lecture.
Hello, my name is Dr. Ashley Beasley Green, and I am a staff scientist at the National Institute of Standards and Technology. And today I will talk to you about biochemical mechanisms of drug toxicity. Here's a brief outline of the presentation. I'll start with the adverse drug reactions, the impact, the economic and the prevalence of these reactions. Some common mechanisms of adverse drug reactions, the type A, the intrinsic, and type B, the idiosyncratic reactions. And also we'll discuss the risk assessment of these drug reactions. The World Health Organization defines an adverse drug reaction as being a response to a drug which is unintended and which occurs at doses normally used in man for prophylaxis, diagnosis, or therapy of disease or modification of physiological function. In the 2014 National Action Plan for the Adverse Drug Event Prevention, which was drafted by the U.S. Department of Health and Human Services, it's noted that adverse drug events account for roughly 2 million inpatient hospital stays annually and roughly a little more than 3.5 million outpatient office visits and approximately 1 million emergency department visits. In addition to the high incidence of adverse events, these events also pose a large economic burden, contributing to roughly $3.5 billion of the U.S. healthcare costs. Therefore, it is extremely important to understand basic drug mechanism of action, but also understand potential adverse reactions that can occur during drug therapy. In this first section, I will highlight several known adverse drug reactions and examples of these reaction types. This is a very simplistic view of drug metabolism. Here we have a drug that's converted to non-toxic metabolites and is then excreted through urine. When bioactivation enzymes convert this drug to reactive metabolites, then we see some of the adverse drug reactions. Listed here are the two major reactions. We have the type A, which is an intrinsic reaction, the type B, which is the idiosyncratic reactions. In the type A reactions, these reactions are dose dependent, therefore they're predictable, and they also can occur at high incidence with low mortality. However, in the type B case, these reactions are not dose dependent, therefore they're unpredictable, and they're dependent on the individual genetic and also physiological conditions, so there is low incidence of these types of reactions. However, these types of reactions occur with high mortality rates. So for the first example, I'll highlight a type A adverse reaction using the acetaminophen drug. This is a well-known and highly characterized drug that is linked to liver toxicity via the formation of reactive metabolites. Acetaminophen is a popular over-the-counter pain reliever, and the mode of action is the inhibition of the prostaglandin H2 synthetase enzyme, which is used to convert arachidonic acid to prostaglandin H2. Acetaminophen-induced liver toxicity is dose-dependent, as with most type A adverse drug reactions. This drug is safe at therapeutic concentrations. However, severe liver toxicity occurs above the therapeutic range. In data collected from the U.S. Acute Liver Failure Study Group in January 2017, It was identified that acetaminophen was the leading cause of acute liver failure. In most instances, ALF occurred as a result of a suicide attempt via a single dose over the therapeutic dose, or from extended use by patients seeking pain relief. These were the unintentional overdoses. In 2015, the U.S. Food and Drug Administration, FDA, issued a final guidance to manufacturers of the over-the-counter drug. Here is the guidance. And it can be located at this web address. Encouraging products to provide alternative language to the liver warning section of the product's label. And here the warning states, liver warning, this product contains acetaminophen. Severe liver damage may occur if you take either one with more than 4,000 milligrams of acetaminophen within 24 hours, 2 with other drugs containing acetaminophen, or 3 with 3 or more alcoholic drinks every day while using this product. Here is the biochemical mechanism of acetaminophen metabolism. We see the detoxification and bioactivation pathways. Approximately 85% of the major metabolites of acetaminophen metabolism are in the inactive forms of the glucuronide and the sulfate conjugates. These conjugates undergo renal elimination, and so then this is the non-toxic pathway of acetaminophen. However, roughly 10% of acetaminophen is then converted through the enzymatic activity of the P450 enzymes listed here to produce a reactive metabolite, as illustrated in this very simplistic view of drug metabolism. This reactive metabolite is NAPQI. NAPQI is then further detoxified by glutathione conjugation. This conjugate is then formed into a cysteine or mercapturic acid adduct. These adducts are then eliminated in urine. In doses higher than the therapeutic dose, a known mechanism for drug-induced liver injury can occur via oxidative stress. In this case, the reactive metabolite, which is the NAPQI, accumulates and glutathione is depleted, which prevents the production of the cysteine and mercapturic acid adducts, leading to urine excretion. So in the absence of sufficient glutathione, the NAPQI reactive metabolite covalently binds to liver proteins. This binding to the liver proteins inhibits the function of these proteins, leading to further oxidative stress, increasing oxidative stress, and liver cell necrosis, leading to liver toxicity. Although acetaminophen liver toxicity is dose dependent, there are several factors that can enhance APAP susceptibility, causing liver toxicity. Alcohol consumption while taking acetaminophen can increase the risk of liver toxicity by increasing bioactivation of the acetaminophen molecule. When ethanol is present, it becomes a competitive substrate for the P450 enzyme illustrated here. This stabilizes the enzyme, which then induces the production of more of this enzyme. When more of this enzyme is present, it increases the production of the reactive metabolite, the NAPQI, further inducing the liver toxicity effects of acetaminophen. So this is why the FDA illustrated in their guidance to reduce the consumption of 3 or more alcoholic beverages in the presence of acetaminophen, because liver toxicity is induced under these conditions. Although acetaminophen liver toxicity is dose dependent, there's also acetaminophen can also induce an immune response. As you can see here, there are several interconnected mechanisms that can lead to liver cell necrosis, liver toxicity following the administration of acetaminophen at certain levels. Unlike type A, type B adverse drug reactions are not dose dependent, and the mechanism of liver toxicity is not linked to the mode of action. Idiosyncratic means specific to an individual. So these adverse reactions are unpredictable. Idiosyncratic drug reactions can affect any organ, the most common of these organs being the liver, skin, and also blood cells. Therefore, liver toxicity is unpredictable. It accounts for a large percentage of drugs eliminated during the preclinical testing, clinical trials, and also the post-marketing phase of the drug discovery and development process. Because it's unpredictable, there is limited mechanistic understanding. There are several theories determined via animal models for type B induced liver toxicities. And I will discuss 6 of these 8. They're highlighted here with the underline. As previously discussed in the acetaminophen example, in the reactive intermediate hypothesis, the drug is converted to the reactive intermediate via bioactivation enzymes and then can bind to liver proteins, eliciting a drug-induced liver injury. The reactive metabolite can also increase oxidative stress, leading to mitochondrial dysfunction, which can then lead to liver dysfunction and toxicity. Also, genetic variations of these bioactivation enzymes can affect the conversion of the drug to reactive metabolites, increase or either decrease the production of these reactive metabolites. An example of these 3 hypotheses is the Isoniazid. This is the first-line agent in the treatment of active or latent tuberculosis. The mode of action is the inhibition of mycolic acid formation, which is an essential component of mycobacterial cell wall synthesis. An animal model was used to determine the mechanism, and approximately 20% of treated patients developed elevated liver enzymes and bilirubin. The reactive metabolite in this mechanism, or reactive intermediate in this mechanism, is the hydrazine. Hydrazine is then converted through the cytochrome P450 enzymes to the toxic metabolite, which then increases oxidative stress, leading to liver toxicity. N-acetyltransferase, NAT2, illustrated here and here, acts at several points to reduce the concentration of the reactive intermediate hydrazine. However, genetic variations of this enzyme can decrease the activity, leading to an increase in the concentration of the reactive intermediate hydrazine, then increasing the liver toxicity effects of this drug. Another hypothesis in the type B adverse drug reactions is the Hapten hypothesis. In this hypothesis, the drug or reactive metabolite, as seen here, the drug or its reactive form, covalently binds to a protein, forming the drug-modified protein. The drug-modified protein then acts as an antigen and is processed and presented on the antigen-presenting cells via the MHC2 in a MHC2 restricted manner. You can see that here. The antigen is subsequently presented to T cells, which can elicit immune tolerance. However, in the danger hypothesis, the similar cascade happens once the drug-modified protein is formed. It is presented by the HPC. However, 2 signals are required in order to activate the T cells, eliciting an immune response from the drug. An example of a drug that forms drug-modified proteins and can elicit an immune response is halothane. Halothane is a volatile general anesthetic administered via inhalation. It was introduced in the 1950s and has been associated with two types of adverse liver events by different pathways. The first pathway, this is type 1 here, presents mild liver toxicity. Halothane undergoes reduction by the cytochrome P450 enzyme illustrated here to produce a radical. This radical can form stable metabolites, it can inactivate the cytochrome P450 enzymes, or most importantly, it can initiate lipid peroxidation. And it is the lipid peroxidation that can elicit the mild liver toxicity effects of this particular pathway. The type 2 pathway, or the severe liver toxicity pathway. In this pathway, the mode of toxicity is thought to be mediated by an immune system response via the formation of a drug-modified protein. So here in this particular pathway, halothane undergoes oxidation by the cytochrome P450 enzyme illustrated here to produce a reactive metabolite that can either form the trifluoroacetic acid or can be conjugated to liver proteins, eliciting an immune response, as illustrated in the previous slide. Unlike halothane, penicillin-induced adverse reactions, or the anaphylaxis reaction, is a classic example of a hapten. Penicillin is an antibiotic, and the beta-lactam ring illustrated here is the chemically reactive component of this molecule. Unlike many other drugs that require metabolic activation in order to elicit a response, penicillin covalently binds to proteins, generating the drug protein conjugate. And it is this conjugate that is the major antigen for the IgE mediated allergic reaction. There are several tiers of reactions that can occur from this drug protein conjugate. The common reactions, such as diarrhea, nausea, rash, occurs roughly greater than 1% of the population. However, the true anaphylaxis response only has an incidence of 0.01%. In order to test an individual for sensitivity to penicillin, the skin test is administered. A positive reaction suggests that the individual should avoid penicillin. This is indicated by the presence of IgE antibodies to the penicillin compound. If needed, desensitization therapy can be conducted further. However, if a negative result is generated from the skin test, it indicates that the person is not allergic to penicillin, or we are unable to come with an actual definitive result. So therefore, further analysis is needed. And one of those is the graded dose challenge. In this challenge, the individual receives 4 to 5 doses of the penicillin molecule, starting with a small dose and increasing to the desired therapeutic dose. If the patient reaches the therapeutic dose and there is no reaction, then that drug is administered and deemed that the person does not have an allergic reaction to the penicillin molecule. Another hypothesis for a mechanism leading to type B adverse reactions is the inflammatory stress hypothesis. An idiosyncratic adverse drug reaction is initiated in this type of hypothesis when a drug therapy is coupled to an acute inflammation episode. It is thought that the inflammatory episode could lower the threshold for toxicity, therefore rendering a toxic response at the safe and effective dose. Examples of these inflammatory stresses could be infection, a disturbance in the intestines, or some type of cell death. An example drug of this type of hypothesis is Trovan, TVX. This is a broad spectrum antibiotic, and it was restricted in 1999 and withdrawn in 2001 due to the severe liver toxicity. It has been shown in mice models that modest inflammatory stress renders safe doses of TVX toxic. This is the mode of action of TVX on the bacterial DNA. In the gram-negative, the drug interacts with the bacterial DNA gyrase. In the gram-positive organism, the drug reacts with the telomerase 4 enzyme. And so actually what happens is the drug binds to the A component of the gyrase enzyme, therefore preventing the separation and religation of the double-stranded bacterial DNA during replication. So this prevents the replication of the bacterial DNA during the growth and production process. In this inflammatory drug interaction mouse model, which was developed by the Roth group, and here you can see the reference for the data presented in this slide. An inflammatory response is induced by lipopolysaccharide, LPS, in the mouse. And then the mouse is treated with TVX following the LPS exposure to determine the effect of the drug on liver toxicity in the presence of inflammation. So this is really to prove that TVX administered in the presence of an inflammatory stress condition can elicit liver toxicity. And here we see ALT actually is a biomarker for liver health. So that's how we're measuring liver health, using the ALT marker. And the vehicle is just the sterile saline material. So in the saline system, we don't see an increase in ALT production or liver injury. However, as we administer the TVX in the presence of the LPS or the inflammatory stress condition, we see an increase in ALT concentration, leading us to suggest that at about 80 milligrams per kilogram, there is liver injury with the administration of the TVX molecule in parallel with an LPS-induced inflammatory response. And over time, we can see here that roughly about after 9 hours of exposure to the LPS or inducing the inflammatory response, we see an increase of liver injury or an increase in ALT here at about 9 hours. So what does this really mean, and how can we really associate this increased liver toxicity to this inflammatory response in the presence of this drug? So the scientists here have looked at the tumor necrosis factor alpha, which is TNF alpha. TNF alpha is a mediator of inflammation and is involved as a major factor in various liver injury models. So it's a good marker for inflammation. So what we see here is the vehicle and also the vehicle with the TVX in the presence of the LPS. So TVX administered during an inflammatory response can increase the TNF alpha. So there definitely is liver injury associated with this inflammatory response. And we can see that here by looking at ALT. So in the presence of TVX and LPS, we see an increase of ALT and an increase of TNF alpha, meaning that the liver injury is induced by the inflammatory response associated with the administration of TVX and LPS. However, we see a reduction in TNF alpha or inflammation, and also ALT or liver injury in the presence of the etanercept drug. Etanercept is a specific inhibitor of TNF alpha activity. So therefore, we're able to reduce the inflammatory response. So as we reduce TNF alpha, we also reduce the inflammatory response in the presence of the TVX. So we can conclude that with this mouse model, when TVX is administered with an inflammatory response, which is induced by the LPS, we see the liver injury. Further confirming the inflammatory stress hypothesis. In addition to the mouse studies, the team also did gene expression in order to identify pathways that are upregulated in the presence of the TVX and LPS system. The findings were that several pathways associated with inflammation were upregulated during treatment with the TVX molecule. I want to highlight here that in the analysis of drug candidates during the drug discovery process, gene expression could be a potential mechanism in order to identify drugs that can elicit adverse reactions, or these type B reactions that are somewhat unpredictable in subjects, especially with acute inflammation. So with all of this information about the type A, type B adverse drug reactions, how do we develop safe drugs? To address the high incidence and also the economic impact, risk assessment strategies must be integrated earlier in the drug discovery and development process. Extensive safety testing is conducted during the preclinical phase. However, to reduce attrition during this phase and also increase patient safety during the clinical trials and the post-marketing phases, the risk of drug candidates should be addressed during the basic research and drug discovery phase. So the earlier that we can identify candidates that can elicit adverse reactions, the safer the patients will be. And we won't spend as much money because this process is extremely expensive. Here in 2013, the costs were estimated to be roughly $2.6 billion for this entire process. So to have a target go all the way to post-marketing and then be restricted, that is a huge economic loss. So we want to be able to enhance patient safety, but also reduce the risk of losing money by a drug inducing some of these unpredictable adverse reactions. Hazard identification and risk assessment of reactive metabolites is one way that we can identify candidates that are pose potential hazards to patients. The major difficulty of identifying these drugs that can elicit these type B reactions is the complexity of the mechanisms involved and also the gaps in our scientific understanding of these drug-induced type B reactions. Illustrated here is an adverse outcome pathway. You have the chemical insult, the macromolecular interaction, cellular, tissue, and the organ response, and then the individual response. So these are different tiers of drug responses. This is a nice representation of the various approaches that are currently being utilized to identify drug targets that can elicit these reactions. Formation of reactive metabolite approach, the metabolite formation combined with a dose approach, and also combining the reactive metabolite to measuring the cellular response approach. So we're looking at these first three tiers of the adverse outcome pathway. The first way or mode to assess the risk of potential drug candidates is to remove structural motifs from the chemical library that can elicit or form these reactive metabolites. The elimination of these candidates based on in silico assessment of structure motifs reduces cost because no synthesis of the compound is required. This approach minimizes risk of developing a drug that can generate a reactive metabolite, therefore leading to patient toxicity or patient injury. However, by eliminating these structure motifs, we reduce the chemical library of for drug candidates. For example, if the phenyl and thiophene structural motifs were eliminated from the chemical library, many drugs that are safe and effective would not be accessible to patients, such as Abilify, which is our antipsychotic therapy, or Cymbalta, which is used to treat depression, or Singulair, which is used in the maintenance treatment of asthma and also to relieve symptoms of seasonal allergies. So we can't eliminate all of these potential toxic structural motifs, but we can eliminate some. Another approach in identifying the formation of these reactive metabolites is a trapping approach. Because of the instability of some of these metabolites, we need to stabilize the metabolite in order to characterize and detect them using these trapping agents. And detecting the trapping agent drug conjugate using liquid chromatography, mass spectrometry approaches. This is an example of the trapping approach used to identify the primary intermediate of the glycolysis pathway, which is the 1,3-bisphosphoglycerate, or 1,3-BPG for short. In the pathway, glucose is converted to GAP under the enzymatic activity of the GAP-DH enzyme. GAP is converted to 1,3-BPG. This metabolite is extremely labile. It simultaneously will convert, or non-enzymatically will convert, to the 3-PG or 2,3-BPG metabolites. Therefore, in order to identify the 1,3-BPG metabolite, we must find a way to stabilize this component without it undergoing hydrolysis or isomerization. One approach that a group decided to utilize the trapping approach in order to capture the 1,3-BPG molecule. Hydroxylamine reacts with 1,3-BPG, forming a stable metabolite that is then that we're then able to identify using mass spectrometry. This stable analog has a unique tandem mass spectrometry profile, therefore allowing the identification of therefore indirectly allowing the identification of the 1,3-BPG metabolite. Here we see the 1,3-BPG metabolite without any modification, and we're unable to detect using mass spectrometry. However, when the 1,3-BPG is trapped or conjugated to the hydroxylamine trapping agent, we're able to identify the modified metabolite using mass spectrometry. And the removal of the phosphate group generates a nice peak at the 199 to 98, and then the 79 precursor product ion transition of 199 to 79. So we're able to detect the modified molecule here. To assess the in vivo detection of the analog or the conjugated 1,3-BPG molecule, the group looked at cell culture. They modified the cell culture with the trapping agent and then just added DMSO as our control. And then extracted the 1,3-BPG unmodified and modified versions and analyzed using mass spectrometry. Here in this bar graph, we can see that the unmodified version in the control system compared to the modified versions have similar relative levels. However, when we add the NAF molecule, which inhibits the activity of enolase, which leads to the accumulation of 1,3-BPG, we should expect to see an increase in the BPG metabolite. There's a slight increase over in the NAF system compared to the control in the unmodified version. However, in the modified version in the NAF system, we see a huge increase, a significant increase in the modified BPG metabolite. So therefore, modifying the metabolite, we get a more accurate view of the concentration of the BPG metabolite. Here we see the mass spectrometry spectra of the modified versus the unmodified in the presence of the NAF molecule. And we don't see the peak in the unmodified version of BPG, but when modified with a trapping agent, we see a peak representing the modified metabolite. They also looked at the concentration of the modified metabolite with increasing glucose. And we can see here that as you increase your glucose, you increase the amount of the modified BPG versus the unmodified metabolite. So what does this all mean? We can actually utilize trapping agents in order to detect highly unstable reactive metabolites, therefore allowing us to determine the formation of these reactive metabolites from drug candidates early on. There are other methods that also can be used, such as the electrochemical approach and also the time-dependent inactivation approach, in order to identify these highly unstable reactive metabolites. The information obtained from the detection of these reactive metabolites can be used to eliminate these drug candidates or structural motifs that can form these reactive species. However, the detection of reactant species does not provide information about the major mechanism of action. Therefore, more work is required to assess the major and minor reactions during drug metabolism. So a reactive metabolite may form, but through these approaches is unable to determine if this is a major or minor form of the mechanism. Also, currently, the assessment of reactive metabolites using these approaches is not compatible with the high throughput platforms, which are used during the drug screening process. Therefore, these approaches cannot be used in these high throughput settings during the early stages of drug development. In addition to the formation of these reactive metabolites, we can also combine the formation with the actual dose. Incorporating the drug dose component to the detection of these species, and also incorporating the formation of the drug protein conjugates is important to identify toxic drug candidates. However, due to the complexity of the toxicity mechanism and in vivo biological interactions that occur with the drug, it is difficult to establish cutoff values that would distinguish safe versus unsafe drugs at early stages in the development process. To address the complexity of the toxicity mechanism, we can couple the identification of these reactive metabolites with cellular responses. One mode to do that is utilizing a multifactorial approach. This can be used as a prediction tool in order to distinguish between drugs that have a high or low tendency to cause these type B adverse reactions. In this approach, we combine the covalent binding burden, which is the quantitative estimate of the ability of the drug to form a drug protein conjugate, with in vitro panel. The in vitro panel consists of a series of assays that are used to profile the biological effects of drugs. This is an example of this multifactorial approach. Here we have an integrated in vitro hazard matrix that's used in order to detect the molecules that elicit no hazard or have an extreme or severe toxicity. In this particular experiment, 36 drugs were screened in this in vitro panel, and also the covalent binding burden was analyzed in order to generate a score that is then used to plot this hazard matrix. And out of the 36, 27 were deemed severe or marked idiosyncratic ADR concern. So they had severe toxicity or teetering on the no hazard and concern level. And 9, it showed a no hazard result. So this particular approach can distinguish between safe and unsafe drugs with high sensitivity and specificity. So unlike the previous approaches where we were just identifying the reactive metabolite or identifying the reactive metabolite coupled to dose, in this particular approach, we're actually combining the formation of this reactive metabolite and also the effect of the drug on various in vitro components. The type B adverse reactions are complex and unpredictable, and in some cases, they can involve the immune system. Therefore, to model these complex systems, we need models that show the interconnected mechanisms that can occur within the tissue or organs. So these existing approaches, although here we're looking at some of the cellular responses, we don't get to capture the inner connection or interrelated mechanisms associated with these tissues and higher order components such as organs. But in this microphysiological model, we're able to capture some of these complex biological interactions and the effect that the drug has on these interactions in order to identify candidates early on in the drug discovery process that can have toxicity to individuals. A few examples of these models are listed here. You can have a 3D culture or liver culture model. You can have a cold culture. You can utilize bioreactors or media flow. I'll highlight one of these models in order to demonstrate how utilizing a more sophisticated model to identify drug toxicity at some of these lower or basic research levels would enhance the determination of candidates that are safe and so those that are not safe to individuals. In our previous example, we looked at TVX. TVX is a known antibiotic, and here we see the mode of action that was discussed earlier. The mode of action for liver toxicity was identified utilizing an animal model where they utilized LPS in the presence of TVX and showed that we can elicit a liver toxicity in the presence of an inflammatory stress condition. However, what if we can use an in vitro model in order to determine inflammation-mediated drug toxicities? In this particular approach, this group developed a 3D liver microtissue, the Kelm group. And it's called the 3D human liver microtissue, or the 3D Insight human liver microtissue. The nice thing about this approach is that it's in a 96-well format. So it is conducive to the high throughput screening process that occurs in the drug screening or basic research process during the drug discovery. This is a 3D liver microtissue that has been converted to the high throughput format. The tissue is stable and functional over 5 weeks in culture, as illustrated in these graphs. We see the ATP, quantification of ATP, and the albumin secretion. So we're able to look at cell vitality and also functionality over 5 weeks. So they're confident that this particular molecule can show long-term effects of drug treatment. So here we have data that utilizes this 3D human liver model in order to demonstrate the effects of TVX in the presence of inflammation on liver injury. And we can see here that as we increase or as we expose the microtissue to LPS inducing inflammatory response, we can actually and also administer the TVX molecule, we actually increase or decrease the threshold of the drug in the presence of the inflammation episode, inducing the liver toxicity. So this is a nice model that provides the ability to identify drugs that elicit these adverse reactions early during the drug discovery phase. So with this particular model, we're able to look at some of the human biological mechanisms that can contribute to drug toxicity instead of conferring an animal model to what could happen in a human subject. In addition to 3D models, we can also utilize computational models in order to provide a cost-effective and safe way in order to screen compounds for toxicity by simulating complex processes during drug metabolism. So in addition to making these molecules and ensuring that we have the correct and optimal environments for growth, and then ensuring that we can actually see a response after drug administration, we can do some computational models. And the nice thing about the computational models is that we can actually generate some of these mechanisms that we can't generate in vivo in order to really capture what occurs after a drug is administered. And we can do this early on in the drug development process. So there are several biochemical mechanisms that occur through drug toxicity. And I've highlighted several that can lead to drug toxicity. And I've also talked about some emerging technologies that we can utilize in order to minimize the risk of drug toxicity. So instead of waiting through the preclinical or clinical, or at the worst case, waiting to a drug is removed from the market during the post-marketing phase, we can utilize some strategies such as the 3D microtissue model or some of these computational models in order to investigate drug targets during the basic research phase to determine which of these targets have the potential to elicit some of these adverse drug reactions, therefore enhancing the safety of patients in the clinical trials and also patients taking the therapies. The key theme throughout this presentation is the need to identify drug toxicity early in the developmental process in order to reduce the risk of to patients during, as I mentioned, during the clinical trials and also the post-marketing phase. So there are a number of mechanisms that can contribute to drug toxicity. And one way to reduce the risk is to start early in the development process, utilizing gene expression or 3D model, microtissues, or computational modeling in order to identify potential targets that can elicit adverse reactions. So I would like to leave you with this thought. I hope that the information that was presented here in these slides will give you more information about some of the mechanisms involved in drug toxicity, but also start you to think about some potential strategies that can be implemented in order to reduce the risk to patients, but also maintain the effectiveness of drug therapies. So the idea is to enhance the drug therapy, but also reduce the risk to patients. And we can do that by looking at some of these models early on in basic research that can more effectively model the human systems, and allowing for a more detailed and in-depth ideal or basis for drug toxicity in humans. So I want you to kind of think about what are some current approaches that can be applied and what are some new approaches that can be developed in order to reduce the cost of drug discovery and enhance the risk or reduce the risk to patients in these drug therapies. Thank you for your attention, and thank you for taking time to look at some of the information I provided here in the slides. Again, my name is Dr. Ashley Beasley Green, and if you have any questions or comments regarding the material that I provided in my presentation, please direct all of your questions or concerns to the program coordinator. Again, thank you, and start the conversation about how we reduce risk to patients by utilizing some of these scientific approaches to identify drugs that can elicit adverse reactions early on during basic research and analysis. Thank you.